Okay, thank you very much for the invitation to talk here. So I'm going to talk about E. coli virulence, but at a broader level, at a population level here. And I'm going to present work that have been done mainly by Thibaut Moret-Journel, who, was a postdoctoral researcher with me, together also with Julie Marin, who used to be a postdoctoral researcher as well, Judith Burgaya-Ventura, who is a PhD student, supervised by Marco Gardini, and also Eric de Namur. And I'd like to thank funding sources, which were first the CNRS and now the ERC. So I'm going to talk about E. coli, which Olivier talked a little bit before. So E. coli is really a specialist of the human gut. It can be found in all of us. It can also persist in the environment. So for example, the sand water. It can also be found at lower densities, lower prevalence in farm animals and wild animals. So it's really a commensal, but we know much more about the infections that it causes. These infections are really rare. They are of mainly two types, the intestinal infections. So E. coli is one of the main causes of diarrhea in children in low and middle income countries. And what we experience a bit more often in Western countries is urinary tract infections, which are quite common and are often caused by E. coli, and bloodstream infections, which are rarer but are quite a deadly form of infection. And altogether, these infections cause a burden of an estimated, this is really an order of magnitude, 1 million deaths worldwide each year, even though it's a commensal, a good commensal. So as Olivier mentioned a little bit, E. coli is a very structured bacterium. So there's several, at a broad phylogenetic level, several phylogroups which may have evolved for of the order of hundreds of thousands of years. And they are called A, B1, B2, C, D, E, F. And for example, A is considered a good commensal. It rarely causes infections. This is the original Escherichia strain that was isolated was a phylogroup A. And B2 is a very pathogenic commensal. A very pathogenic phylogroup, and it is the one that is responsible for most extraintestinal infections. So the human and non-human animal strains are largely distinct. So this is a phylogenetic tree that was obtained from strain samples from various sources in the UK, and mainly the two sources were livestock and human. So livestock human in orange here, and livestock in green. And you can see on this tree that for the most part, the diversity is really clustered, and there are strains that are found in humans, like this large block here, and strains that are found in livestock. And even if you zoom in on areas of the phylogenetic tree where it's all a little bit more interspersed, if you zoom in on this little clade here, and there's a lot of mixture, but if you do the calculations of how many years of evolution separate these strains, it's actually, so you can do that with a molecular clock given the number of SNPs, so it's quite convenient because one SNP difference is approximately 1 year of evolution in the species. But anyway, so clades like this one and the neighboring clade that is more found in humans are separated by maybe hundreds or thousands of years of evolution. So it's really rare that you have successful cross-species transmission. So you can, I think it justifies that you can really study the human strains by themselves. So E. coli is a really important pathogen. There's a very sharp increase in the incidence of these bloodstream infections with age. So the points here are coming from this meta-analysis. So each point, each series of points comes from different studies in Western countries. And there is a quite robust pattern whereby in the 80+ individuals, there's almost a 50-fold increase in the incidence of bloodstream infections compared to the young individuals. So if you project the demographic dynamics and how this will change the incidence of bloodstream infections, there will be, from now to 2050, there will be almost a 2-fold increase in the incidence of infections in countries like those of the European Union, and it can be even sharper in countries where there is a faster aging population. And so this is projections, but this is also shown in data that the incidence of these infections is increasing fast, which will make E. coli one of the most important infectious diseases in a few years. There's also the problem of antibiotic resistance, which makes infections harder to cure. And E. coli, according to a large analysis that was done at a global level, E. coli is a pathogen for which there will be the largest, or there is already the largest number of deaths associated with resistance to antibiotics. Fortunately, there's a vaccine that is being in development for extraintestinal infections. There was a phase 2 trial published a few years ago. So this is testing the safety and immunogenicity of a vaccine that is targeted at the serotypes that are most commonly found in E. coli causing infections. And now there is an ongoing phase 3 trial, so testing safety, immunogenicity, and also effectiveness of the vaccine on thousands of volunteers, which are elderly individuals who are most at risk of urinary tract infections and bloodstream infections. However, we did not have a very clear view of, or systematic view of the bacterial genetic determinants of pathogenicity. So this is the first part of my talk, where we worked together with Julie, Judith, Marco, and Eric to try to really identify what is the bacterial heritability of pathogenicity. So what fraction of the variation in pathogenicity is attributable to bacterial genetic factors. And to do so, pathogenicity is defined as the probability to cause an infection. And there's a long tradition of trying to identify such factors by comparing bacteria sampled from the gut of healthy individuals and bacteria from the blood of the blood or the urine of infected individuals. And this has been done for decades, but it's also often quite small studies. People often actually sample the stool of the individuals that were infected, and this can considerably limit the power of such studies. And also, people often just test a handful of genes that they suspect might be involved, but they don't have whole genome sequences. So here, what we did is first we had this really large and quite rare sample of E. coli genomes from healthy volunteers in Paris. It's surprisingly difficult to actually access or to get volunteers to give stool samples, which is why this collection is quite rare, compared with close to 1,000 genome samples from infections. This is what the data look like. This is phylogroup B2 here in red, and you can see, so the circle in red and green represents the status infection or commensal. So you can see there's lots of infections in this red circle here, and phylogroup A in green, there's much more green here. So as I said, it's more commonly found in commensals. And we also have quite good metadata on these strains, which is important to control for potential confounders. So the first finding is that we can systematically characterize the pathogenicity of different sequence types. So sequence types are clones that are like a finer phylogenetic scale than phylogroups. And we find a very large diversity with many quite pathogenic sequence types, often belonging to phylogroup B2, but not only, and also some sequence types that are circulating in the commensal population that are not very much pathogenic. And this hierarchy is largely conserved when you focus on the urinary portal of entry, so the people who had a urinary tract infection that degenerated into a bloodstream infection, and similar ordering when you consider the digestive portal of entry, which is people who got a bloodstream infection directly from the gut. And perhaps our main finding is that pathogenicity is actually quite heritable, so we have close to 70% heritability. So as compared to, if you compare it to previous GWASs of people investigating the pathogenicity of various bacterial species, it's really on the upper end of the spectrum, so a species with a lot of bacterial genetic factors that control pathogenicity, and it's similar to, for example, Streptococcus pneumoniae. And an interesting finding is that, so this is for the full collection. This is the heritability that is generated or contributed by all genetic variants, and the sequence type lineages is only quite a small fraction of this heritability. So you have the sequence type effect is quite a small fraction. So we have a lot of heritability that goes on top of this clone effect that can be attributed to individual genes or individual SNPs. So what we did next is that we did a genome-wide association study, and the goal of the study is to try to find measures that predict pathogenicity. And the data really looks like a matrix like this, where the rows are the bacterial strains, which are pathogenic in red, and the commensal in green. And you characterize each strain with a genetic variance, and we use a unitic variance that allow to represent both the gene content and also the individual SNPs. And there are hundreds of thousands of these genetic variants, so there's much higher dimensionality along this axis than the number of strains we have. So what we are doing to predict pathogenicity is a lasso regression, which is like a linear regression, but you try to choose which genetic variants are most predictive of pathogenicity by penalizing the effect size that you can give to each variant. So basically, you're just getting rid of all the variants that are unimportant, and you can select just the ones that are contributing most to the predictive power. And the result of this algorithm is that we identified dozens of SNPs that are predicting pathogenicity, and so we have a measure of predictive ability, which is actually the S1 score, so how well you predict the status, commensal, or pathogenic of strains. And so we are looking at this predictive ability in the subset of all the phylo groups. So the gray point is what the performance you have if you just choose at random whether it's a pathogenic or a commensal strain. And we are looking here at the improvement that we can have with our algorithm. So the naive algorithm is without covariates, and the yellow, the blue point is the predictive ability without the covariates, and the yellow is with covariates. So for example, within phylo group B2, with the genetic variance, we don't improve much prediction ability, and I think this because B2 is very pathogenic, so you have a lot of, if you just do by chance, you just say, well, it's going to be a pathogenic, and it's not so bad. And then within other phylo groups, we can actually improve quite a lot. So for example, within the A phylo group, we can improve quite a lot our prediction of whether this particular strain was sampled from an infection or from healthy individuals. So the variants that we retain in the GWAS are, so as I say, there's dozens of them, and it's enriched in classical or non-virulence genes such as adenosine or iron capture systems, but there's also genes that don't have like a non-function that could be investigated further. And finally, we did something a little bit different. So we have this predictor of pathogenicity and commensalism, and we applied it to all commensals of our data set. And we have this additional data set that we didn't use for the GWAS, which is composed of commensals sampled in 1980 in healthy volunteers. So we are looking at what the algorithm predicts on all these commensals. So whether it predicts correctly that they are commensal, in green here, or whether it predicts wrongly that they are bloodstream infections. And when we did that, what we found is that there was an increase in the predicted pathogenicity over time, from one time period to the next and then again, which means that the strains over time resemble more and more bloodstream infections rather than resembling commensal, which might hint that there is an increase in pathogenicity over time in France. And this is to be taken with a pinch of salt, because even though it's reassuring that we find this jump in the two time periods, I didn't find any other data set on other countries where we could find these, investigate whether we find this strain. So there's no other data set of commensals over a long time period that could be investigated further. So to conclude on this first part, so we found in E. coli a large heritability of the capacity to cause extraintestinal infections, and this goes beyond just the clones, the sequence types. There's a lot of variability that is explained by genetic variants that are not organized along these sequence types. The portal of entry was actually quite important to control for. we have this lasso method that pinpoints tens of variants associated with infection, and possibly pathogenicity is evolving upwards in France. And these lists of variants could actually be used for better and more durable vaccines, because if you target directly the variants that have a causal role in infection, you expect to get rid of the disease and avoiding possible rebound in the strains that are pathogenic and not targeted by the vaccine, as was observed for the vaccine against the Streptococcus pneumoniae, for example. So the second thing I wanted to present today is interested in the epidemiological dynamics of E. coli phylogroups, and this is work that Thibault Morel-Journel did. So just to remind you, this population structure. So the question is, why do we have incommensuralism, a coexistence of all these phylogroups? And people don't really ask this question often, so they don't really ask about the epidemiological dynamics of E. coli in the population. And one of the reasons is that we don't have this like regular longitudinal sampling of healthy volunteers that we will need to address this question. There's been a handful of studies since the 1960s, often with like 5 to 10 people followed for some time, but there's no large systematic such data set. However, recently, researchers in the USA published a really nice data set on 8 healthy volunteers who were sampled for 1.5 years to 2.5 years at quite regular time intervals. So this is showing the time series of one volunteer here. And these people went to really, they did a lot of work to characterize the diversity of the dominant E. coli flora. So the y-axis shows different clones. So they characterize, I think, up to 30 clones per time point. And therefore, they have quite a good view of the diversity within each individual. And a typical profile looks like this, where this individual is colonized by this strain for a few months. There's occasional diversity, and then there's succession dynamics where other strains are seen at regular intervals. So many strains are seen only once, and some others are more persistent. So this is the whole data set on the 8 volunteers. So there's the one I just showed is here. There's people who have more diversity or more like clones that can persist for a longer time. And there's also like this person here where this person is colonized by 3 clones that persist for extended periods of time together and apparently are quite robust to the colonization of other strains. So this data set has been published, and we wanted to really use this to get an epidemiological description of the strain turnover and what is the epidemiology of E. coli in this community. We formulated a mathematical model of host colonization by E. coli, which is really similar to classical SIS model in epidemiology. So we describe people who are not colonized, which are not many, colonized by, for example, strain A or strain B, or they can be colonized by 2 strains of type A, 1A, 1B, etc. And this can go on. And we actually describe, we have a model that can describe arbitrary number of colonizations. And the nice thing with this model is that we can parameterize it and infer directly the parameters from the data. And namely, we have these rates. Sorry, I didn't describe the arrows, but so you have 2 types of event: clearance of the strain and colonization by a new strain. And we define clearance of a strain of phylogroup K within a host carrying already N clones, among which NK are of the same phylogroup K, by this term here. So essentially, this is the mean persistence time in the host, that's the denominator here. So there's a phylogroup effect, so how good is this phylogroup at persisting? A competition effect, so how other strains within the host affect your persistence time. And there's an additional effect of competing with the same phylogroup. So what difference does it make whether you're competing with other strains that are similar to you or different from you? And similarly, for the colonization rate, we define the rate of colonization of phylogroup K in a host carrying n clones as a baseline times the factor that describes the ability of the phylogroup to colonize, and also an effect of competition. And I mean, we inferred this as a term here, but we found it has no, this has no, it's inferred to be 0. The main finding from this analysis is that the diversity of E. coli that is found in these volunteers is structured along a continuum from the strains, for example, the ones of phylogroup B2.3, which are really good persisters, so they can persist on average for 2 years in hosts, but they are really poor colonizers, so you don't see them very often arriving and colonizing a new host. On the opposite end of the spectrum, there are strains such as this phylogroup A.1 here, which are not persisting for very long, maybe a few days on average, but have a really high colonization rate. So you see them popping up over and over again in the dataset, but they don't last for very long. And in this model, the fitness of a phylogroup, so its selective ability, is actually given quite simply by the basic reproductive number, which depends on the colonization rate by the phylogroup times the colonization time. So it's a quite simple result, but it was not so simple to obtain because we have this diversity, this multiplicity of colonization, so it was not immediately obvious that it would be the good measure, but it is. So we have quite simple terms that describe how good is a phylogroup, and it turns out that along this line, all phylogroups have similar fitness. So really this trade-off between persistence time and colonizing ability enables all phylogroups to have similar fitness and potentially to coexist. And in addition, we also infer that this effect of competing with the same phylogroup is significant, and this means that individuals who have 2 clones of the same phylogroup will have larger clearance time of this phylogroup compared to people who have a diversity of phylogroups, which means that you have an additional mechanism that stabilizes diversity. So from this model, where all parameters were inferred from these data, we were able to predict E. coli colonization dynamics and the dynamical consequences of what we found. So this is showing here for an example of 2 strains competing together in a host population. So this is the fitness difference between the 2 strains and the niche differentiation term, so how much more you compete within your phylogroup compared to other phylogroups. So the data suggests we are around here, and so the color code is a measure of diversity, so how much diverse will be the E. coli population. Basically, in the blue zone, it means one of the strains win over the other, and here you can maintain some diversity. So we are in a range of conditions where the diversity of phylogroups can be maintained. And the importance of this finding is also that we find that the phylogroups which are persisting for longest are the ones that are most pathogenic. So we have 2 measures of pathogenicity here. So one is the odds ratio of infection risk as derived from the GWAS that I presented in the first part. And the persistence time correlates very strongly with this pathogenicity. So with B2.3, the most pathogenic one being here and A1 being here, for example, but it's true for all phylogroups that you have this organization. So the strains that are most pathogenic are the ones that are able to persist for longest, but they're also the ones that are really poor at colonizing new hosts. Another measure of pathogenicity is indirect, based on the mean number of virulence genes, and there's also a correlation at this level, and the category of virulence genes that actually correlates the most is adenosine. So it makes a nice link with other talks, for example, the one from Alexandre. So what we find at the population level is that the strains that have the most adenosines are the ones which can persist for longer, but they're also really terrible at colonizing new hosts. We can also derive predictions of what could favor the more pathogenic persisters. And what we find is that if you reduce transmission rate, for example, you don't alter the competitive hierarchy between different phylogroups. However, when the baseline clearance, so which you can think of like the baseline rate of perturbation of the host microbiota, so when you tend to reduce the level of perturbation, you tend to favor the persister pathogenic type. So this is shown here as an example, with 3 phases where in the model, we reduce the level of external perturbation, and the persister types increases in frequency, and again, with an additional level, like additional reduction in perturbation, it can also win over the colonizer type. So this is because the persister type has more, it has more to win by reducing the level of perturbation, because this strategy is based essentially on persisting for a long time. And finally, we could use this model to derive predictions on the impact of a vaccine. And for example, so if you apply a vaccine that is targeted at a virulent type on maybe like 10% of the population that is most at risk, and this vaccine is very efficacious, you predict that you will decrease the R0, the basic reproductive number, by about 10%. So basically, you can move along this axis, increasing the fitness difference between the different phylogroup, and you can increase it such that the pathogenic phylogroup is predicted to disappear entirely, which means that the vaccine could have an impact beyond just the direct protection of people who are immunized, but also by disturbing the whole dynamics of the level of population and actually getting rid of the pathogenic type entirely. So the conclusion on the second part is that we formulated and fitted to data this mathematical model of E. coli epidemiological dynamics. We find that this broad structure into phylogroups that has persisted for hundreds of thousands of years in the human population might be explained by the fact that there's a diversity of strategy from persisters to colonizers who actually have similar fitness. There's some evidence that there's niche differentiation within the host between these phylogroups. It's important because the persisters are actually the most pathogenic strains. And we've seen, for example, in trends that the pathogenic strains have increased in frequency, or may have increased in frequency. We predict that less perturbation of the microbiota would favor the pathogenic persisters, and that an E. coli vaccine that could be distributed to the population in the coming years could actually possibly get rid of the pathogenic types entirely, even if it's applied only to a small fraction of the at-risk population. That's all I wanted to say. Thank you for your attention.